Hey guys, welcome back to my channel, and today we're going to do a quick review of Unit 2 in the AP Biology test, which is about the cells. Okay, now this is a book, Barren, and for me it is very useful for reviewing for the AP test. So without further ado, let's get right into it. So first question is going to be, which of the following is correct about G proteins, which is shown in the diagram below? And here, I'm going to zoom in the diagram for you. As you can see, the G proteins are labeled here, alpha, beta, and gamma. And as you can see that here it is in its inactive state. And when the ligand, and you can see here in the active state, the ligand bond with the G protein coupled receptor on the membrane, and the thing is that it triggered the reaction to happen. And as you can see that the G protein bonded with the enzyme and it's changed in its shape and then it released this substance called CAMP, and which will trigger the cell response. So you can see that let's take a look at some options. The first one, it is a membrane-bound receptor, which is not true because you can see that it's not a receptor, right? The receptor is actually the thingy here. I'm pointing on it, and it's actually a protein that was triggered by the reactor or the receptor. Okay, so A is incorrect. It is a cytoplasmic receptor that bonds to hydrophobic substances, which is also wrong. There's no indication about whether the substance is hydrophobic or hydrophilic. It is an on-off molecular switch that gets activated by the membrane-bound receptor. Makes sense because you can see here that the ligand, when it is bonded with the receptor, it triggered the action. So C might be an correct answer. It is an ion channel receptor protein that is the basis of normal neural function, which is also wrong because this is not what's actually happening in the neuron cell. And as you can see that here we got nothing indicating that it is a neuron cell. Next, which figure depicts an animal cell's placed into a solution hypotonic to the cell? So I'm going to take a look. What is a hypotonic solution? And as you can see that a hypotonic solution will have a lower concentration of solid than the cell. Right? This is opposite with hypertonic solution, which has higher concentration. So here, what happens if we put a cell inside a solution that is hypotonic, which has a lower concentration of solute? And you know that the water, right? The water will flow from high to low concentration. So the low concentration of solutes means that the high concentration of water. So the water will flow from the outside to the inside. So here we have a solution, and the water will flow from the outside to inside the cell. And look at these diagrams. You can see that only B makes sense, right? So the answer is B. I cannot highlight the B, so just let me type in here. This is our answer. Signal transduction pathways are complex, involving many different molecules and many steps. What is the main advantage of sending a signal through a signal transduction pathway to an opposed sending a signal through contacts between two cells? So okay, signal transduction pathways are complex, right? And you see that there are some ways to transduct informations. So you see that here I have two adjacent cells, and this is this direct contact between two cells, right? Contact, right? It's very quick, to be honest. But however, for signal transduction, it would be like when a ligand is bond with a receptor, it will trigger a chain reaction, right? This is a cell response, right? With many steps. So actually, it's not going to be quicker or faster, right? So, and there actually might be more chance for error since it requires many steps. So we can already eliminate the answer A and D, right? We can eliminate A and D. Okay, next. Direct contact messaging is limited to simple cells, like bacterial cells, which is incorrect because direct contact messaging is actually evident in plant cells and animal cells too, right? So it's not true. So we only left with the answer B. Signal transduction pathways offer more opportunities to amplify the signal, which makes sense because with all of these steps, right, extra steps, you have many more chances for the signals to be amplified. Right? Actually, just from one single ligand here, molecule, it can trigger a chain of reactions that will cause the production of many molecules, right? Bisphenol A is an organic synthetic chemical that has been used in the manufacturing of hard, clear plastic. It has been used in manufacturing of plastic water bottles and baby bottles, as well as to line drinking water pipes. The EPA has ended iteration for the use of EPA in many products, and the European Union can error. This is the same. Actually, you don't need to care about this whole long text because it should only give you like the background information that is actually not required in the test. Then, however, you need to look carefully at the diagram right here and to see what the BPA do to the body. So here we have the BPA, which looks strikingly similar to the estrogen hormone, right? This is cytosol, which is the environment surrounding our enzymes, right? Our receptors. Okay, so here is the estrogen receptor. So normally in a healthy body, the estrogen hormone will bond with the estrogen receptor. So they will trigger the chain reaction in the cells to produce some molecules that will help with the normal living activities. However, the BPA is strikingly similar to the estrogen, so it can bond with the receptor too. Therefore, less estrogen hormone are received, so therefore it will negatively affect the chain reaction of the body. Okay, so that's its danger, and now we're going to take a look at all the answers to find out which one makes the most sense. BPA converts estrogen into another substance and blocks normal estrogen pathway. It doesn't seem correct to me because it doesn't react with the estrogen in a diagram, right? BPA prevents estrogen from binding to the G protein receptor and blocks the normal estrogen signal transduction pathway, which is actually not really true because you see that the BPA bonds with the receptor, but it doesn't prevent this estrogen specifically to bond with the receptor. BPA combines with estrogen and diverts estrogen's normal function. It's also wrong because it doesn't combine with estrogen here. You cannot see that in diagram. BPA competes with estrogen for its membrane receptors and subverts normal estrogen function. What does subvert mean? Subvert means to decrease the function. Okay, so D is the correct answer here, right? Because you see that it competes, right? Less estrogen hormones are being able to bind with the receptor, right? So the normal estrogen function will be subverted. Okay, next. which of the following statements is correct about apoptosis? So first, what is apoptosis? So apoptosis sometimes is called programmed cell death. Okay, apoptosis is also called programmed cell death, which means that when a cell has some problems, maybe some defects in its genes or its function, and it would be harmful if the cell keeps on dividing. So the body will, it will trigger apoptosis, which is the process that will make the cells die instead of dividing, so that it would not be harmful to the body, right? So here, let's take a look at some other choices. First, the process of apoptosis has only been identified in advanced animals, such as tadpoles. First, we're not going to consider tadpoles as advanced animals, right? Actually, it is present in tadpoles, but however, the apoptosis is also present in plants as well, right? Not just animals. Plants also have apoptosis, and it's actually very crucial in the development of plants. It normally triggers an aged cell to undergo mitosis, which is wrong because mitosis is a cell dividing process, right? And here, the cell is going to be dead. There's no dividing happen, so it can't be. It is an important part of normal embryonic development. Which is true because you see that the embryo, for example, when we're in our mother's belly, you see that we have a little tail here, right? This is the human fetus, right? And actually, our tail dies. It's cut off, and it is owed to this process of apoptosis, and it's very crucial in the development of the embryo, the fetus. So C is the correct answer. And let's take a look at D, just in case we might be wrong. The signals that trigger the process always come from outside the cell, which is not true because this can be triggered by both inside and outside factors. Right. A plant cell is placed into a beaker that contains a sucrose solution. The setup is shown in the diagram below. Okay, so there are cell, and here is a water potential. Okay, so which of the following is correct about what will occur and gives the most accurate reason for the occurrence? As you can see that inside the cell, it actually has higher water potential than the sucrose solution. So, as you can see that something with higher water potential, the water will flow from inside the cell to the outside of the cell because in inside there is higher water potential, right? So when the water flow from the cell to the outside, it will not swell, it will shrink. So C and D is wrong. And now we're going to look at the reasoning to find which one makes sense. The water potential is greater inside the cell, which is correct. So our correct answer is A. You tag amino acids with radioactive sulfur because amino acids are incorporated into proteins in the cell. You monitor the location of these tagged amino acids until they are released from the cell. Which of the following best describes the movement? So first, okay, nucleus cookie backfill, and now you need to look at the production of amino acids, right? So okay, first, production. What will the cell turn amino acids into? It will turn into proteins, right? And so here it is the production of protein. So basically, we're just going to track the amino acids and how it will be turned into proteins and what will be its pathways. And here, the first proteins is synthesized by the ribosomes, right? But wait a minute, ribosomes aren't called organelles, and you can see here which organelle here actually has ribosomes. So as you can see that Golgi apparatus doesn't have ribosomes. Nucleus, no. Vacuole, no. But you can see here that the ER actually has a ribosome. It's specifically the rough ER. Endoplasmic reticulum has ribosomes, so it is in the rough ERs, and we can just shorten it into the ER. And then our protein is going to be what? To be packed, right? It needs to be packed before delivering, so it's going to be inside the vesicles. It's going to enter. Oh, it looks like sockets, like this. So this is the Golgi apparatus, right? And after that, it will be packed into these vesicles, and then it's going to travel to the plasma membrane, and then it'll be exit out of the cell to function, to aid with the, just to do its biological function. So the pathway will be from the rough ER to the Golgi apparatus to be packed, and then it will be transported to the membrane, and that is where our amino acid will travel. So here you can see that C is going to be our correct answer. Right, it's exactly that pathway. Some pancreatic cells produce insulin, a polypeptide that consists of 51 amino acids in great abundance. Which organelle would you expect to find in pancreatic cells in greatest number? So you see that it produces insulin, right? And that insulin, a polypeptide that consists of amino acids. So insulin is a protein, okay? So it has lots of protein. So we need to find an organelle that is often attributed to proteins. First, vacuole is not related to protein. You see that you can easily choose ribosomes, right? Because ribosomes is where you will synthesize the protein. Okay, it's not going to smooth endoplasmic reticulum. You know that actually the ribosomes in the rough endoplasmic reticulum do the job, not the smooth endoplasmic reticulum. It rather had things to deal with the production of lipids, right? Okay, here. Let's get to the next question. Which of the following is correct about microtubules? They make up flagella in Paramecia. They assist in the formation of pseudopods in amoeboid cells. They consist of actin fibers, and they assist the formation of the cleavage furrow in animal cells during cell division. And so here we actually need to, there's two things, right? Microtubules versus microfilaments. So here you can see that microtubules actually make up the flagella and flagella in the cilia in bacteria. So the flagella is going to be long telomeres, and cilia is going to be many short hair-like structure. And for microfilaments, it will do with the hair formation of pseudopods in amoeba, right? This is pseudopods, right? This formation is actually the job of microfilaments, and also it will contain actin fibers, and also they will assist in the formation of cleavage furrow in animal cells, which is when you have two cells here, it's going to create a cleavage. And that's also the job of microfilaments. So you see that this is actually some theoretical stuff, and you need to review that in your books, in your textbooks, right? So our answer is going to be A. There's no logic inside this. You need to memorize it, So if you cannot do this question, I would recommend you to review that in your textbook and make sure that you will get the hang of it. Right? You need to know the difference between microtubules and microfilaments. Right? So let's take a look at this. So I'm gonna dive a little bit more into the microtubules because they actually do another job, which is the formation of spindle fibers. You know that in cell divisions you got like the cells like this is the centrioles, right? Okay, then these are the spindle fibers. You got your chromosomes right here, right? It looks like meiosis. Okay, you got chromosomes right here, and they pulling the chromosomes like two ends of the cell. So the filaments right here is actually the spindle fiber. And this one is made out of microtubules, right? So when you think of microtubules, you think about these hair of bacteria like the flagella and the cilia, and also the formation of spindle fiber, and that's it. Everything else have something to do with microfilaments, not these guys, right? Just remember to know the difference between these two kinds of fibers because they can be really confusing for you. Okay, next, Dr. Lynn Nicholas. We don't need to care who she is or he is. Developed the theory of endosymbiosis. This theory explains how prokaryotic cells came to exist, eukaryotic cells came to exist, circles across the membrane against gradient and ligands enter cells. So the endosymbiosis actually, basically how it's basically explain how in animal cells or in plant cells there's like a mitochondria inside it. So yes, you know that we have these guys, right? Mitochondria, and also this little guy too, the chloroplast. These guys actually can be considered as an prokaryotes, right? Prokaryotic cells, and they just enter inside the animals. For these guys, it would be the plant cells, right, with an actual nuclei. So that is a theory of endosymbiosis. So it's actually going to explain how eukaryotic cells came to exist, right? It does not have anything to do with ligands or sucrose crossing the membrane against the gradient. Feel that. This one has something to do with the transportation of substances through the membrane, and this one actually has something to do with the signal transduction process inside the cell, and from the outside to the inside of the cell. Right. Let's take a look at question 11. Refer to the diagram below. Which of the following choices describes which cell, A or B, would be most efficient in terms of transporting nutrients and waste across the plasma membrane, and provides the correct reasoning for the answer. So for this type of question, we're going to need to pay attention to the equation here. So this is the area to volume. Sometimes it can be written like this too, because it's actually the surface area of volume. So if this ratio is high, then if the ratio is high, then it would be easier to go for the metabolism actions, and also it is more efficient in terms of transporting nutrients, just stuff moving from outside inside of the cells, right? So here, let's take a look As you can see that in the cell A, in the smaller cell, the ratio is much higher, right? Right? Because you see that the ratio here is actually... Okay, if you take a little bit of math into it, you can see that it's actually something like this. So it is inversely proportional to the radius. So the smaller the radius will, result in higher ratio. So this is like for suppose spherical cells, but it is like the same for most of the cells, right? So here we have the cells is much smaller, so it will have the higher ratio of the membrane surface area to the volume, right? So we can easily choose A. I don't even need to find out what is choice C, because I already know that A is correct. So we're going to choose A in this case, right? Next, below is a diagram of an amoeba engulfing the bacterium here. So as you can see that it forms pseudopods, right, around the bacterium in order to swallow it. And as we have stated before, that the pseudopods, the formation of pseudopods is aided by the microfilaments, right? So this is the guy that we're going to pay attention to. Okay, amoeba contains which of the following is true of an amoeba as opposed to a cell that does not move, right? Amoeba contains a larger nucleus? No, of course not, because whether the nucleus is large or small, it has nothing to do with the mobility of the cell, right? So A is not the correct answer. A is not correct. An amoeba contains more microfilaments. Well, this is true, because the microfilaments is responsible for the formation of pseudopods, right? Responsible for the formation of pseudopods. So B is correct answer. It contains more microtubules. No, because microtubules has nothing to do with the formation of pseudopods. As you can see, and that pseudopods help with the mobility, right? An amoeba contains more sensory apparatus, which is not true, because actually you can see that there's no sensory apparatus here. It's a little, right? It's just a cell. Below is an illustration of an important cell organelle. Okay, so you see here that we have this is definitely the Golgi apparatus, right? You can see that there's like flattened sacs. So the Golgi apparatus has two faces. And you can see here that right before it would be this ER, and the side of facing the ER would be the cis face, right? Closer to the ER. And the trans, opposite to cis, obviously, which is further from the ER, right? The first is not a rough endoplasmic reticulum. I see. Here. Okay, first we're going to look at a diagram to, just analyze this a little bit. You can see that it has a cis phase, right? Incoming transport vesicle. And here, it's going to go into these sacs to form new vesicles that we called secretory vesicles through the trans phase. So this is not a rough endoplasmic reticulum. We don't see any ribosomes here, so it's obviously not a rough endoplasmic reticulum. It is not even an endoplasmic reticulum, because for endoplasmic reticulum, it's not going to be sacs, flattened sacs like this. It's going to be like a funky maze of, just these things, just funky. It looks like a maze, but it's not, it only have like it doesn't looks like a sac. Here, the smooth endoplasmic reticulum is producing proteins in attached ribosomes. No, smooth endoplasmic reticulum doesn't even have ribosomes. B is false. Clearly, like today, this is a Golgi body that is pipetting material sent from the endoplasmic reticulum near the cis phase, which is true because that's actually what we're seeing here. That the incoming transport vesicles here is going to be packed into these vesicles, right? And it's coming from the cis phase to the trans phase. So D is pretty much pretty similar to it. It's just that it states that endoplasmic reticulum is near the trans phase, which is false because in here in the diagram we can already see clearly that the incoming vesicle is near the cis phase, not the trans phase. So C is our correct answer. Which of the following would not normally diffuse through the lipid bilayer plasma membrane, and give the reason for your answer? So to diffuse into a lipid bilayer plasma membrane, it is actually going to be something that is. So what can we diffuse to through a lipid bilayer plasma membrane? You can see that a lipid bilayer is actually not fully hydrophilic or hydrophobic. Is what's the word for it? Just let me check my dictionary real quick. Okay, so you can see that amphipathic. Yeah, you're right. That's amphipathic. Oh my gosh. Let me think. Yes. It's never mind. So it means that actually if this molecule is small enough, it can easily diffuse through the cells, whether it is hydrophobic or hydrophilic. Okay, I can just get it out. It's been bugging me. Okay, so CO2 can easily diffuse through the plasma membrane, and you can see it clearly in the red blood cell, right? Water can diffuse easily through osmosis, and you already know that, right? That's why we have the experiment with these kinds of experiments, like you cell in a solution, whether it is hypertonic or hypotonic. You see that's happening all the time because water, despite being polar, it can obviously diffuse easily through the membrane because it is simply way too small, right? Starch, because it is too large. This is our correct answer because it's too large. It cannot normally diffuse through the layer. Amino acids, because it is acidic and charged. No, it can still diffuse through a lipid bilayer because if it's small enough, it gets through. And moreover, acidic and charged has nothing to do with the diffusion, right? Through the membrane. It's it's actually the matter of the concentration, right? Normally it would not be, these kind of things. Which of the following require ATP? So we're looking at something that needs require energy, something active, right? So here, the uptake of cholesterol by a cell. So the uptake of cholesterol by a cell, the facilitated diffusion of glucose into cell. So the facilitated diffusion is actually still a passive diffusion, passive transport. It just has like a protein channel for it to go through. Countercurrent exchange. Countercurrent exchange is also a form of diffusion, right? It's just, increase the efficiency of it. Diffusion of oxygen into fish gills also is passive transport from high concentration to low concentration. So we have obesity, is all passive and does not require ATP. And we are left with A, the uptake of cholesterol by ourselves. And so what is exactly the process for this? And it is because cholesterol is actually a very large molecule, right? So it needs something to facilitate its transportation. So it needs a special receptor to bring it into the cell, right? So if you don't know, you cannot, if you feel that reasoning is not convincing enough, you can always take a look at obesity and realize all of those processes like B, C, and D, it's about diffusion, and it has nothing to do with the energy, right? Because it's passive, it just happens. You don't need energy to induce it to happen. Which of the following best characterize the structure of plasma membrane? And you can see that plasma membrane rigid and changing? No, obviously not. Rigid but varying from cell to cell? No, absolutely not, because plasma membrane is actually not rigid at all, and it is made of. Okay, so the plasma membrane is made of these phospholipid bilayers, right? And you can see here there are cholesterols to just consolidate it a little bit. And here we have proteins, or maybe some receptors, and still phospholipid. This is actually very active, right, and organized. It's not chaotic at all, right? Everything has its own function in here. So unorganized, no, it's not unorganized, and it's not actually fluid, right? It's like semi. So I'm going to choose D because of all of the transportation of substances inside and outside the cell happens all the time, right? We have ions, we have water, the transportation just happens all the time. So you see that it's very active indeed. Below is a diagram to plant cells. Okay, cell wall, cytoplasm, vacuoles, you see that it's a direct contact, right? So the gap at X, what role does it play? So actually, for these kinds of gaps in animal cells, we're going to call it a gap junction. But for plants, it's gonna call plasmodesma, right? So this is a plasmodesma allow for rapid passage of water and small dissolved solids from one cell to an adjacent cells, right? This is obviously not the xylem in here. You can see that the cytoplasm is traveling across them, and the reason that I choose this is because it's called plasmodesma, not a gap junction, because the gap junction is a word for animal cells, right? So it's about cell communication, and that's about cell communication. And if you cannot do this question, I would recommend you to review that part. You can just Google it or look it back in textbook. But basically, for those direct contacts, types of cell communication, so it's called gap junction in animal cells. And for plants, those are called plasmodesma for plants. Okay, that's it. Refer to the figure below, which shows the plasma membrane, and obviously it's which the following correctly matches the part with this function. A is involved in facilitated diffusion. It was not true, because facilitated diffusion will be our protein channel, and A is actually something that looks like the receptor. B is cholesterol, which is false because you here see that it's phospholipid. So C is the lipid, the two fatty acids. I'm sorry, C are the two fatty acids, and B is the phosphate group. The head of the phospholipid. Part C allow oil-soluble substance to freely enter a cell. D is an ATP synthase channel. You can see that C is going to be our correct answer because these are fatty acids and like dissolves like, so oil-soluble substances will be able to freely enter a cell due to these fatty acids. Which of the following is prokaryotic cell? Human red blood cells? No. The bacterium E. coli that lives in the human intestine. Of course, not human bone cells that are eukaryotic, plant cells that are eukaryotic. B is the correct answer. Human red blood cell, it doesn't need to have nucleus, right? However, it does not have, like genetics materials at all. So that's not going to be the case. Below is an illustration of plasma membrane transporting two substances, sucrose and protons. And you can see here, this is a co-transporter. And in here, you can see that sucrose is entering through passive transport because it is entering from high to low concentration. However, for sucrose, it's going from low to high concentration. So you see one active and one passive. Sucrose is diffusing down its gradient through a channel. It's not true because it's the one going down the channel here is down the gradient here is actually protons, right? So A and B is wrong. You can easily eliminate those. Sucrose and protons are simultaneously being pumped against the gradient. This is not true because protons actually being pumped along its gradient. So that's left with C. Sucrose being carried against a gradient only while protons are diffusing down a gradient, which is exactly what I've been discussing with you. Which of the following statements about cell-to-cell communication is correct? Plant cells cannot communicate with neighboring plant cells because the presence of thick cell wall, which is incorrect because they have here these two, right? That's a plasmodesma. Proteome sensing enables bacteria to monitor the population's density and to use this information to control gene expression, which is true because it is true. But don't worry if you don't know about this, you can always eliminate C and D in order to arrive at the answer. And let's take a look at C and D. Hydrophobic chemical messengers, estrogen, rely on a secondary messenger cyclic amp to initiate a response from the nucleus. Well, this one is not true because estrogen can actually dissolve through the plasma membrane, so it doesn't rely on the secondary messenger. Regardless, so here actually you need to at least remember the truth, right? You need to remember that estrogen can actually dissolve through the plasma membrane. Regardless of the type of chemical signals, the first thing that happens in receiving cell is transduction, where the chemical message may be converted into cascading signal in the transduction pathway. It's not true because the first step is the bonding of the ligand with the receptor, right? Okay, we're arriving closer and closer to the end of our practice. This illustration below shows a glass tube that contains two different solutions of sugar separated by a selectively permeable membrane marked with an arrow, which allows for the diffusion of glucose only. Okay, so it only allows glucose to be diffused through a membrane. See here that which of the following choice is correct to describe what will occur and give supporting evidence for that occurring. So you can see that here the overall concentration of sugar is higher in B, right? So you see here that the water potential of side A will be higher than side B because the concentration of sugar is higher than B. We have 3 actually 3 moles per liter here, and here we have 1 plus 1.5 is 2.5, but 3 is larger than 2.5. So you can easily see that B is our correct answer. Glucose will diffuse from side B to side A because molecules flow down a gradient. It's not true, because actually in side B, you see that there is less glucose, right? It's not going to be down the gradient. There will be no net movement because concentration of glucose is greater on side A, because glucose is greater on side B. But it's not true because it's not, greater at the same amount. There will only be no net movement if the water potential on both sides are the same. Glucose will diffuse from side B to side A. It's not true because this selectively permeable membrane doesn't allow for sucrose to diffuse from one side to another. Which of the following is correct about signal transduction pathways? Are found only in the cells of the most complex animals? No, it's also present in plant cells. Signal transduction pathway have evolved recently along with the development in large brain size in mammals? No. Once again, it is present in plant cells. In the signal transduction pathway, a single molecule can stimulate with the release of thousands of molecules of products. This is actually what we're talking about, right, within a cell. And that's the amplifying function of this transduction process. Signal transduction pathways are unique in receptors as stimulate the pathway located only on the surface of plasma membrane. No, because it can happen. Not only on the surface of plasma membrane. Right. Which of the following initiates a signal transduction pathway that leads to response in the cell? And obviously it's going to be what? It's going to be... No, it's not cyclic AMP forming cytoplasm. Can you transfer the ATP to another molecule inside the cell? No, ion channels open. And it's obviously a receptor changes its conformation because it is bonded. Because it is activated, right? The receptor gets activated. This is bonded with a ligand. Which of the following is correct about chemical signals that bind to receptors are located on a cell membrane rather than receptors in the cytoplasm? They are hydrophobic, they require a second messenger. Nitric acid is one example, and of course not. They're oil soluble. As you can see that. Located on the cell membrane. So you get the receptors in the cytoplasm, right? Inside the cytoplasm, there's a cell. There's a receptor inside the cytoplasm, so it can trigger the activation of the process inside the cell immediately without needing a second messenger. However, on the outside of the cell, you need to trigger like a receptor here. Here's our receptor, right? You need to trigger a second messenger, which is here is our second messenger. Right, we will induce a series of reactions inside the cell. So that's located on the cell membrane. Will need the second messenger. Right. Which of the following is correct about receptors that are located inside the cytoplasm? So they don't require a second messenger. It's not the ion channel receptor because this one is obviously on the cell membrane. They bind to hydrophilic substances? No. They bind to signal molecules in the cytoplasm, which causes a response in the nucleus. Is what they would do. Signal molecule in the cytoplasm, right? Which of the following most accurately describes the advantage of a signal transduction pathway? Yes, it can occur in the cascade effect. Well, obviously, the binding of signaling by cascade effect, but it does not actually state that is an advantage. I cannot see what's good about it here. right? It needs to be further elaborated. The binding of signaling molecules causes receptor protein to change its conformation. And so, that's not actually an advantage, that's not clear. Binding signaling molecules can happen on either the surface of the cell or cytoplasm. And here, one binding molecule can trigger the release of thousands of molecules, and it's exactly the amplifying advantage of the process. Which of the following correctly describe the role of cyclic AMP? It acts as a second messenger for signaling molecules can pass through the cell membrane. This is actually what it's going to do, so it's a correct answer. But if you cannot understand why, the reasoning behind this, you can always choose to eliminate the other choices like B, C, or D. Let's take a look at B. It assists in the activation of the tyrosine kinase receptor. No, it's not. It activates the G protein coupled receptor. It's not, because here, actually, in the first question, you can see a clear diagram here that the G protein will bond with the enzyme, therefore turning ATP into cyclic AMP, right? No, so you see that's not the case. It does not activate the G protein. It's actually like something, opposite of it, like the G protein coupled receptor, right? It was activated by the ligand, not the cyclic AMP. The ligand comes from outside the cell, and the cyclic AMP was produced, right, when things that's inside the cell. It is an allosteric receptor in an ion channel receptor. It's not a receptor, okay? It's not a receptor, it's a substance that don't do that. It's the end of our practice today, and I hope that you guys are having, a better hang at theoretical stuff, the multiple choices questions, the AP biology exams, and here, this is the chapter summary, something that you can review all of it. All cells share some characteristics because they all arise from a common ancestral cell. Eukaryotic cells are particularly efficient because they are compartmentalized by a selectively permeable endomembrane system, which allows for separation of different chemical processes from each other. The fluid mosaic model describes a selectively permeable plasma membrane that separates the cell from its environment, but that also allows for the regulated exchange of materials with the environment. And a sufficiently high ratio of surface area, some membranes to volume, is necessary for exchange of material into and out of the cell. Cells communicate with each other with the environment through complex system reception, transduction, and responses. And that's our main idea. And remember to review that carefully with your textbooks. Okay, so this is the end of our today's studying session, and thank you for watching this video, and I hope you guys will score a high score in the test, and I hope that the same will happen to me too. Okay, so that's the end, and I think I gotta say bye.